I'm going to let your fellow classmates, Jadalyn and Megan, talk more about Dr. Venter in particular and his work, but I want to talk a little bit about why we decided to have you hear from Dr. Venter this afternoon, and what about him makes him a distinguished Mills graduate. So our vision for you is that you embrace learning for growth and that you become lifelong learners that lasts for a long time, even after you're here at Mills. And I think you're going to find Dr. Venter's story a powerful lesson about how and when that growth happened for him and how he was able to use his resiliency and growth mindset to contribute significantly to science and our understanding of genetics. But also, in Dr. Venter's story, I want you to listen for ways that his experience relates to yours. The story of a Millbrae kid who was eager to work with his hands, build and tinker, explore and play. A Millbrae kid who went to the same elementary school, probably, maybe the same middle school, and definitely the same high school that you did. A Millbrae student and a Mills student who was not a straight-A student. He began in school and struggled in math and other subjects, but he did not let his performance define him or his future. He didn't allow being a C student make him sentenced to a life sentence. Yep. A Mills student who went on to serve his country. He experienced the horrors of Vietnam, and he came home a changed man. A Mills student who went first to community college before embarking on his UC path. A distinguished Mills graduate who did more than just think outside the box, and oftentimes contrary to the contemporaries in his field. Dr. Venter defined the box in many ways for his field. Dr. Venter is much more than a man who changed science and our understanding of who we are on a genetic level. He is one of our own. He's a Viking and one of our most distinguished Mills graduates. And I can't thank him enough for coming home to Millbrae to speak to you. It is my pleasure to introduce two of our students, your fellow classmates, Jadalyn Egan's and Megan Campbell, who will talk more about Dr. Venter and his work.
Okay. Dr. Craig Venter is a graduate of Mills High School, class of '64. Dr. Venter was an unmotivated student during his high school years, never being the one who was projected to be on one's most likely to succeed list. He preferred activities like swimming and surfing over his required classes. While he was working as a medic overseas during the Vietnam War, Dr. Venter realized that he wanted to pursue a career in the scientific field. Dr. Venter came back to be one of the first people to sequence the human genome. During his work on the Human Genome Project, Dr. Venter founded Celera Genomics. His company used Venter's controversial method of shotgun sequencing to map out the human genome. Although his method was counter to the scientific methods being used, his method became widely accepted in the scientific community after Celera published their results.
Besides his world-famous work on the human genome, Dr. Craig Venter researched on synthetic genomics and the human lifespan. Dr. Venter is currently the president of J. Craig Venter Institute, which continues various genome research. Additionally, Dr. Venter co-founded Synthetic Genomics, which researches synthetic biology and specializes in clean fuels, and is currently working with ExxonMobil to create better biofuels. Dr. Venter's latest company, Human Longevity Incorporated, is currently working to sequence 40,000 genomes, focusing on cancer genomes and the genomes of cancer patients. Craig's goal is to redefine current approaches to healthcare and thereby extend healthy lives. With that, please welcome Dr. Craig Venter.
Well, thank you, ladies, for the very kind introduction, and thank you, Pam, for hosting us here today. So it's nice to be back here after 53 years. I hope you guys are doing better in your classes than I did. But as you heard, I barely graduated. I got a D- instead of an F in a government class. And the only reason I got a D- instead of an F, the government instructor was a Barry Goldwater fan. So from my final paper, I wrote, even against my beliefs, that Barry Goldwater should be president, and he was so impressed he graduated me. So otherwise, I'd still be coming back here trying to get a degree, I guess. So I'm going to try and put things in context of history, because I like to think about that with, I have some cars and motorcycles from the 1930s, the 1940s, the 50s and 60s, and I try to think of what happens at different times in history. And I've heard that a lot of you are concentrating on science. Obviously not all of you, but many of you are. And you've heard about Charles Darwin. But it's only been 142 years since Darwin's theory until the human genome was sequenced. And some of the most important things have happened in the last 72 years. So perhaps the most important experiment was 2 years before I was born, showing that DNA was in fact the genetic material. Up until then, biochemists had the wrong idea. They thought proteins were more complex, and a simple molecule of DNA could not possibly be the genetic material. And so just this, as I said, 2 years before I was born is when the first realization was that DNA was this material. So a few years later, this is what I was doing in Mowbray. You had to have steel wheels then, but I didn't know much about DNA or history. It wasn't until '53 when the work you've heard about Watson and Crick actually discovering the structure of DNA was known. This was what I looked like in 1953. And this is what my report card looked like from the 7th grade at Taylor. You saw in the introduction, I went to a different school every year. No school wanted to keep me, so they kept moving me around. But I was at Taylor for 2 years. I don't think my high school report cards look much different. I did well in 3 areas. I did well in sports. Interestingly here, I actually did well in science. That was my best grade, a B+. In manual arts, which was wood shop, and I really enjoyed that. You can see some of the other things like spelling, I didn't do particularly well at. In fact, I refused to take spelling tests because I thought it was really stupid to memorize a list of words and regurgitate them the next day on an exam. And they didn't quite know how to deal with that. But in my spare time, and this is a more modern photo than the one I was looking for, but before they put in this part of the runway, this runway here used to just abut right next to this creek here, and so I claim I'm responsible for updating airport security because with a couple friends, we'd go out to this runway on our bikes and we'd race the planes for takeoff for the first little waves. And a bicycle is much faster because planes, you see, start slow. But it used to really freak out the pilots. But the airport terminal was way over here, so you could see the police cars coming because they had to come all the way over here. By the time they got over here, we hopped over the ditch and disappeared. And after doing this for a couple weeks, we came back and there was a fence around the runway. So we started the trend of fencing off airports. So I think that was my first major contribution to society. So I started coming to Mills in 1960. In 61 is when the genetic code was actually determined. I didn't pay much attention to that at the time. This is what I looked at 3 years later in 1964. And I decided I wasn't really interested in college, although I was offered swimming scholarships to a few places, including Arizona State, but I wasn't sure I wanted to keep doing competitive swimming. So I ended up moving to Southern California to take up a surfing career. It was very short-lived because I got drafted. And a long story short, I ended up in the Navy Medical Corps. It changed my view of knowledge. I was like a sponge absorbing information, doing procedures, and I found out that knowledge was power. So I came back extremely motivated to start school over again. And so I started at the College of San Mateo in 69 or 70, and that's when some of you have studied these restriction enzymes were discovered. I was not aware of it at the time, but one of the people that got the Nobel Prize for it, Ham Smith, has been working with me for the last 25 years and is in his mid-80s. I transferred to UC San Diego after a year and a half. And this is kind of what I was doing while I was attending classes. I had a nice little sailboat that I used to sail here on San Francisco Bay and used to sail around San Diego Harbor when I wasn't in class. I got my bachelor's degree in 1972, and that's when the whole field of recombinant DNA started here in the Bay Area. I got accepted to graduate school at UC San Diego, and I took time off to bicycle around Scandinavia, going to a scientific conference. So, I was continuing to explore the world in different ways. But I went from barely graduating from this school to having the shortest Ph.D. from the UC system. Just a little over 3 years. So when you're motivated, even if you have learning disadvantages, I have aphantasia, which is the opposite of a photographic memory. I have no visual memory whatsoever. So I have to work harder for memorization, but concepts I'm extremely good at. So. But the first gene wasn't even sequenced while I was a graduate student. So I got my Ph.D. in 1975. The first genome of a virus wasn't sequenced until 1977. So the word genome didn't even exist when I was a graduate student. I went from UC, I didn't have to do a postdoc. I was given a faculty position in medical school at State University of New York. It was in Buffalo. There wasn't a lot to do there, so I used to race Hobie cats instead of surfing in my spare time. But my career slowly picked up, and in 1984 I moved to NIH, and I had a new idea of combining new sequencing technologies. I had the first automated DNA sequencer with new computing. It was a simple idea, and it's what I want you guys to think about. A lot of the breakthroughs that I'm associated with started as very simple ideas. cDNAs are chemically made from RNA, isolated from tissues. And my notion was, well, why not just isolate a bunch of them and just sequence them using these automated sequencers? And we went from discovering one gene in 10 years to doing hundreds to thousands a day. So it was a huge breakthrough, and it developed all kinds of new mathematics around it. But it's a technique that really pissed people off, because they'd be working for 10 years trying to find this one gene, and they'd find that I randomly discovered it in that day's work. So it didn't start off, being heralded as a breakthrough because it made so many people angry. They actually said it was unfair I was making too many discoveries. A few years later, in 1992, I had the fortune, I was offered $70,000 to start my own not-for-profit research institute. And that seemed like a reasonable thing to do at the time. I was a government employee. I think I had the top salary in the government at $80,000 a year. So, and we continued to sequence human genes, but we got the idea. In fact, it was from meeting up with Ham Smith at a meeting in Spain, and we became fast friends and spent a few days in the bar and at dinner with each other, and came up with an idea to try sequencing the species, Haemophilus influenzae, that he recovered the first restriction enzymes from. And so we tried to get money from the government to do that, and our grants were turned down. They said our method was impossible. But in a short while, we were able to assemble this whole genome, and this is what the very first genome map looked like. Each of these little colored bars represents one of the genes. And the goal was to try and understand a species from the first time in history from seeing all the genes. You can see I had a whole lot less hair then than I did in earlier photos. So, but this was the real start of the genome revolution. And as typical with government funding, well, we couldn't get any funding to do this project. Once we proved this idea worked, we got unlimited funding to do hundreds and hundreds of genomes. We did a second one that year, and it's the smallest genome of a living species. And I'll come back to this later, because this is what started the entire field of synthetic genomics and synthetic biology. We decided we were going to make a synthetic species to really try and understand genomes at the most basic level. But I got a phone call. I got several phone calls. I actually thought they were hoaxes. This company in the Bay Area called Applied Biosystems called, and people said, we want to give you $300 million to sequence the human genome. Of course, I thought it was a hoax. After they tried several times, they finally came out to visit, and it turns out they were for real. And that's how we started Celera Genomics. And for the first test, we set out to, on the way to human, we sequenced the fruit fly, which the genome was thousands of times bigger than the first bacterial cell. So all this work now was making very frequent headlines, and started to get much more known. And sequencing the Drosophila genome was a major milestone, because so many people work on fruit flies that having the complete genome in one step was a very big deal. But what it really proved is the algorithms that we were developing would work. It was less than 9 months later that, in fact, we had the first draft of the human genome. So it was all starting with a small idea of just using mathematics to sort out random sequences, to using random sequences to sequence genomes at a small scale, and then proving that worked with assembly algorithms and faster computing that we could solve bigger and bigger genomes. So to do this, we were competing with a $5 billion public effort. The Human Genome Project was supposed to be a 15-year project. So we did this in 9 months for $100 million. So it's still a lot of money, but it shortened that project by a decade or more. And while it was considered one of the most important scientific achievements, it also made a lot of people working on the other side angry with me. But it ended up with a nice event and a tie where the finishing of the genome was announced in the White House. Now, this is all bringing it up roughly into where you guys were born. I'm sure most of you were born this century. Was anybody born in 98 or 99 here? Okay. Wow. Have you guys been held back or something? It's, in 2000, you were a couple years old, and you were probably not aware of these events, but they started a lot of interesting things happening. So this is a picture of my institute in La Jolla. It's on the UCSD campus. It's celebrating its 25th year this year. And this is the first carbon-neutral research building in the world. Thank you. It's probably hard to see, but the entire roof is a 0.5 megawatt power plant. And San Diego gets enough sun generally to keep it fully powered. The water is recycled with big cisterns underneath, which finally with this rain has been able to get filled. But the research that the Institute did led to the human genome sequencing. But the same idea led us in a number of different directions, and I'll show you just one or two of them. Because we were sequencing microbes with the very first genome, I became one of the most famous microbiologists, even though I never trained as a microbiologist. But we decided to ask, again, some pretty simple questions about life. I don't know if you guys know where the Sargasso Sea is. It's near Bermuda and around Bermuda. And oceanographers and biologists were sort of characterizing microbes in the ocean. And they were characterizing basically one at a time. They got up to SAR 11, so that was Sargasso Sea 11. And it just didn't make any sense to me. So we took my sailboat and went out there and filtered some seawater and sequenced all the DNA from these organisms. And what we discovered is that there were thousands and thousands more species than anybody ever imagined. So this is what we did for 5 years, sailing around the world, taking samples. This is the route that was followed mostly around the equator, so that we hit all the great dive sites in the world, and it was warm water. But we stopped every 200 miles and collected about 400 liters of seawater and filtered them. And each site, just from sequencing the DNA from those microbes, we discovered hundreds of thousands of new species. We extended it into the North Sea and then into the Mediterranean and around, of course, we had to sail around Greece for a very long time collecting samples. And then we went up into the Black Sea, going through the Bosphorus. But here's a basic example in a graphical thing. So SAR11 was thought to be a single species. When we lined up all the sequences we found against the SAR11 genome, we found that basically there were tens of thousands of related species to SAR11, but they all had slightly different genomes. And so this is what we find everywhere, including in your human gut. We use these same techniques, and that became what's now called the microbiome. It's not just single sets of microbes, it's complex families of microbes. So this was published in a special issue of PLOS, an online journal. We got back to sort of the basic question we started with the first genome, and a key assumption about your own lives, studies you may be doing. Does DNA contain all the necessary information for life, or is there something else involved? And people have speculated on this for centuries, even before they knew that DNA was the genetic material. So we decided to go a new way. We decided to start with the ones and zeros in the computer and see if we could regenerate life from this digital information. So the way we did this is we had a machine that used 4 basic chemicals where we could actually synthesize DNA. And when we started out, you could only make fairly small pieces. We started to say, you may remember a little bit earlier, 5X174 was the first DNA virus that was sequenced in 1977. So its genome is only a little over 5,000 letters long, and we decided to try and make that first as a test. But the machines that print DNA make errors, so we had to come up with new ways to correct these errors. And you'll notice that Ham Smith, the Nobel laureate, was an author and has been a key collaborator on this study going forward. So we synthesized the genome and we injected it into E. coli. E. coli immediately recognized this chemically made piece of DNA as normal DNA, started reading it, started making proteins. The proteins self-assembled to form the virus, and once there were sufficient particles there, they lysed the cells. So these clear spots are where the virus killed the E. coli and then went on to infect other cells, all starting with a synthetic piece of DNA software. So we call this a situation where the software builds its own hardware. Now, computer scientists would love it if that was possible. You just write a new code and you instantly get a new computer. But that's what happens in biology. Now, we didn't want to just make small viruses. We wanted to make a complete self-replicating life form, so we wanted to make a complex bacteria. But the tools to do this just didn't exist. So it took us over a decade to develop all these tools to go from making something 5,000 letters to making something over 1 million letters of genetic code. And so we finally were able to do this, and simplistically, we make pieces that are 1,000 letters long and put 10 of those together, make pieces that are 10,000 letters long. And then we put 10 of those together, make pieces that are 100,000 letters long. And then we put these into yeast, and yeast assembles these by homologous recombination. So we ended up with this big circular 1.1 million base pair synthetic piece of DNA in yeast. We isolated it from yeast, and we transplanted it into a receptor cell. And remember the restriction enzymes that Ham Smith discovered. So restriction enzymes cut DNA at recognized parts. So in the genome that we made it so the restriction enzymes would recognize the DNA in a cell. So we transported this new synthetic piece into this cell, and now we have 2 pieces of genetic software. And what happened is the new piece of DNA started to be read and proteins were made. The early proteins were the restriction enzymes that recognized the chromosome in the cell as foreign DNA and chewed it up. So now that we have the body of one species and the genetic software of another. In a very short period of time, we ended up with these brand new cells where every molecule in the cells, every protein in the cell, every chemical was either derived directly from the synthetic DNA that we put in or from proteins made from that DNA. So we described this as synthetic life. In 2010, this was a big announcement and started off this whole new field. And in fact, to identify synthetic life from natural life, we came up with a way to watermark the DNA. We developed a new code where we could write the entire English language with numbers and punctuation in DNA. Now, people had done this before just using the ASCII code, but our code puts in frequent stop codons. So, for example, if you wrote out Mills High School, you might develop a new toxin that would kill people. So by putting in frequent stop codons, you can't get new peptides. I don't know if you can read this. The code gave the way to translate the DNA code into English. We had a URL in there. This code told people if you solve this to send an email to the species. So being the first chemically made species, it was appropriate to have its own URL. It has the names of all the scientists that contributed to the work, the names of the institutions, and then I put in 3 quotations from the literature. The first is from James Joyce, to live, to err, to fall, to triumph, to recreate life out of life. That's what we were doing, so that seemed appropriate. The second one is from American Prometheus, Oppenheimer's biography. He was told by an early teacher in high school, see things not as they are, but as they might be. And the third was from Richard Feynman, what I cannot build, I cannot understand. Now, once this became widely public, one of the first things we got was a letter from James Joyce, a state attorney, asking if we had copyright permission to use that quotation. James Joyce, as you know, has been dead for quite a while. And US copyright laws allow you to use up to a paragraph or so with attribution. So we dismissed that. And then we started getting emails from a Caltech scientist saying we had misquoted Richard Feynman. We argued, if you look up on the internet now, you'll find this quotation. And finally, for evidence, they sent a photo from the Caltech archives of Feynman's original quotation, which I think is much better. What I cannot create, I do not understand. So we don't know if it was his biographer that changed it or how it got modified, but we like his original version better, and we went back and changed the genetic code so Feynman will rest peacefully with the right quote. So I think all this proves that life is a DNA software system, and if you change the software, you change the species. So we've taken this a step further. We wanted to design a species from scratch in the computer. We had several teams working on this. None of the original designs worked, so we had to develop this design, build, test cycle. But finally, we ended up with this new cell that was designed for the first time in the computer. And the surprise was a third of the genes were new to science. So what people thought was the essential biology, we found out that we didn't know. So this is what these cells look like. This was published just this last year. And it turns out these new genes are part of life everywhere, including the human genome. The last thing I'll show you is we're trying to defrag the genome, redesigning it in a modular fashion. It's actually pretty amazing we can do this. We can reorder the genes by function. So if we're going to design species in the future, you want to just pull out the different cassettes to put things together. But this shows you how many different things you have to rearrange just to get the genes in the order of function. Our work's undergone a lot of ethical scrutiny from the beginning. It scares a lot of people thinking about making new life forms from scratch. There are whole studies sponsored by the Sloan Foundation and others. And in 2010, when we announced the first synthetic cell, President Obama asked his new bioethics commission to take this on as their #1 charge. And so if you're interested in this, you can download it from the White House website. At least I hope it's still there now with the new administration. Well, I won't say what I was thinking. So the ethics of this is important. Releasing organisms into the wild is not something we're contemplating, but this work has allowed us to understand life at the most intimate level, and it's affecting very much how we approach the human genome and how we approach human health, something I'll talk more about this evening. So I'll stop there, and I'd be happy to take questions. Thank you very much. There's one right here.
With regards to your cruises on your ship...
Put it closer to your mouth.
My apologies. With regards to your cruises around the world with your ship on finding microorganisms, it said that on your Antarctic cruise you had no data. Could you explain a little why that happened?
So the speakers go out. Say that again?
Okay, can you... on your chart of the cruises you've taken regarding finding microorganisms in the ocean, on your Antarctic cruise it said you had no data. Could you explain what happened there?
So a fiberglass sailboat's not a good boat to go to Antarctica in. So we have teams that go to Antarctica. They've gone there every year for the last, I think, 12 years. And they take samples from drilling into the ice cores. They take it from the various lakes around there. And there's as much, if not more, diversity. That one of the frozen lakes, they found there was higher density of microbes there than in the open ocean. So Antarctica is steaming with life, even though it looks like just frozen ice.
Thank you.
Hi, my name is Michael Chang, and I have a question for you. You mentioned that...
Hello, Michael Chang.
Hello. It's very nice to meet you, and thank you for coming. So I have a question regarding your obstacles and hardships during high school. So you mentioned that you have aphantasia. What other types of obstacles or hardships did you face as a student at Mills?
Well, I got suspended once or twice. But one of the most interesting times, you have another extremely well-known person associated with Mills High School. His name is Gordon Lish, who is a famous author, editor. He's now 85. Many in our class had him for an English teacher. He taught primarily Catcher in the Rye and had a unique, unorthodox approach. And he ended up being fired. And so I helped start a protest that shut the school down for, I think, 3 days, which didn't make the then principal very happy. So. I don't recommend it at all. But you do, we're getting into an era where we absolutely all have to fight for our beliefs, and I started early. That's as far as I'm willing to go on that question. Here's one here.
Hi, my name is Norden. I have a question. So when you went to Vietnam, you said that after you were there, you had a thirst for knowledge and your realization of knowledge being power. Can you explain that a bit more?
Yeah, it's something I'll be talking about quite a bit tonight as well, but the more knowledge I had, the more lives I could save. And it made me thirsty for learning even faster. But I think the reason I was able to do so well in school once I got out of Vietnam, I had a context for all the learning that I was doing. That when you're just learning stuff for the first time and you have no context for it, if you're good at memorization, that works well. But I need a context for things. And so learning how things worked and how that knowledge could go on to change people's lives is a very powerful tool. And in fact, that's what we're doing at Human Longevity, of showing that by detecting diseases very early or predicting them from the genome, we can save people's lives and prevent diseases from happening or getting rid of them early. And while you're all very young, 30% of males in the US will not reach the age of 74. And 20% of females will not reach the age of 74. So I'm 70, so I haven't quite made it yet, so I don't want to brag too much, but I'm getting closer. But 2/3 of the reasons that people don't reach that age is heart disease and cancer. Cancer is higher in women, heart disease is slightly higher than men. And so by having knowledge of being able to detect things, predicting it from your genome, or detecting it with new technology we think we can save lives and prevent premature death. So if you can come tonight, I'll be talking more about that.
Hi, my name is Sarah, and I have a question about how exactly did you deal with criticism and the backlash against moral issues and ethics, and did it ever deter you from your research?
So it's a good question. Most of the criticisms and attacks, well, some of them were superficially based on morals or ethics, were really not based on that. And so, but it doesn't matter what the source of the criticism was. If you read Gene Wars or my book on the genome, it was an epic battle in science. And just like today in politics, scientists aren't so nice to each other, and they use a lot of name calling and other things. And no matter what it is you do, it's a matter of believing in yourself and believing in what you're doing. If you do that, then criticisms can be more of an annoyance than anything else. They don't deter you from what you're doing. And I also had great teams and great colleagues to work with that all believed in what we were doing.
Hi, my name is Annie, and I just have a question. You've been the vanguard of the scientific community for quite a while now, and what future projects are you looking forward to, especially in such an age of progress and innovation? What are you looking forward to in the future?
Well, I appreciate the question. So, most people are long retired by age 70. We started Human Longevity just 3 years ago. So we're just getting into the most exciting stage of science and medicine. With the change in computing, we and all kinds of other people, you read it and everything Google and Apple and Amazon are doing is applying machine learning to complex issues. But we're applying machine learning to everything from understanding the genome and understanding medicine, so that by the time you finish medical school, if that's what you decide to do, medicine and science are going to change to be totally data-driven disciplines that, if you're a physician, you will help guide the use of that data. whereas now physicians get data to help them do what they're doing. Machine learning and the richness of the data that we and others are generating are going to change the entire future because we're going to have so much more absolute knowledge at our fingertips. And that's going to happen in the next decade. You guys are going to be the ones to benefit the most from it. And you're at the time for probably the most exciting ride in science because the changes are going to be so radical in a very short period of time. There's some in the back too, and some over here.
Hi, my name is Holman, and I wonder if you think that synthetic life have a limit, or if there's no limit, what would you say that the most dangerous trait will be for synthetic lives?
So, making synthetic life is like any other tool, any other part of science. It can be used for trying to create new ways to manufacture medicines, new ways. It could be the new industrial revolution, making chemicals, making everything we can do, or people can try and make things to cause harm to people. So it's not a new dilemma in science or in life, but it's a new, much more complex one, because nobody's ever had the ability up until now to actually design new life forms. But on the encouraging side, it's actually extremely difficult to do. We had a large, very qualified team that took us almost a decade to design a very simple bacteria. So I'm not worried right now about people designing new pathogens, but it's like anything. It's over here in the back.
Hey, what's up? I'm Nick Voss.
Where are you?
Right here.
Okay.
Hey, I have a quick question. What part of Millbrae did you live in?
What part of the what?
Where in Millbrae did you live?
On Laurel. When I was on the good side of the tracks, I lived on Laurel Avenue. When I was on the other side of the tracks, I lived on Beverly Avenue. So.
That house in the picture, it looks familiar. Have a good day.
I can tell you how to climb out the second story window. It's in my book. It describes how to do that.
Hi.
Where are we?
My name is Hosea. Okay. What is one thing you would like us to remember from your experiences in achieving what you did?
What would I like you to remember? Not to be limited by dogma, and find something that you're passionate about, and you can believe in yourself, and you can believe in what you're trying to do, and do something to make your life worthwhile. If everybody does that, we'll solve the problems in society that are so rampant right now. Yes, sir.
I believed you skimmed this over a bit before, but I wonder what hurdles are in the way of us and creating genetically engineered babies, and I want you to repeat more about your moral stance on it.
So you would like to make a genetically engineered baby? Is that what I understand?
I just want you to say if there's anything.
Okay. It's
that's
much more
between
fun
us.
to do it the natural way. It's, But to seriously answer your question, even though that was a serious answer, we are just at the earliest stages of understanding the human genome. I said we're about at the 1% level of knowledge. So we're not at the stage where we understand enough to start editing it. And even though CRISPRs sound like a nice tool, they have a lot of off-target effects. So you think you're changing one base in the genetic code, and they're actually changing a lot of others. And if we start using that to modify embryos, we could be creating new diseases, new defects. And if people do that prematurely before we understand the science, it could get legislations and rules passed to block it forever. So we need to wait at least until we have sufficient knowledge to do it intelligently. And it is more fun to do it the natural way. There's questions.
Hi, Matt. You talked a little bit about machine learning and like data analysis in traditional science. I was like wondering, like, do you think bioinformatics is gonna be like the next big tech boom kind of thing, like the next big industry?
It already is, young man. Over half our employees are bioinformaticians, algorithm specialists, machine learning specialists. So if you're looking for a job, come check with us. It's the most exciting field. Tonight I'll show you data on how using machine learning we can predict your face, your photograph, right from your genetic code. So it's some of our first machine learning results. There's a couple here in the middle. I don't know where the microphones are. Uh-oh. They've cut you off.
Okay. I want to thank you for your questions. Excellent questions, Vikings. And I want to thank Dr. Venter. We have a short presentation. I want to bring to the stage a partner who has been working with me on this presentation, and actually part of the class of 1964. There's two other classmates that initiated this, wanted to bring Dr. Venter back to Mills to speak to you and do a short presentation in honor and recognition of him. So I'd like for Jadalyn and Megan to come join me out on the stage, and I'd like to welcome George out as well.
Hi, everybody. I'm not a brilliant scientist, but I am a Mills graduate of 1964. and grew up here in Millbrae, just like Craig, and we were neighbors. But it's my privilege now to recognize Dr. J. Craig Venter for his amazing accomplishments in science. And on behalf of the class of Mills High School 1964, we'd like to present this plaque, which will be on stage within this, somewhere on the campus, for Dr. Venter.
All right. Thank you very much. Thank you.
I want to thank you for this plaque, and Dr. Venter, please know that you will continue to be recognized and have your rightful place here on the Mills campus. And thank you, and thank you for being a distinguished graduate. I'd also like to make sure to give you a Mills sweatshirt. Students, do you think he has earned his red, gold-blooded sweatshirt this afternoon coming out to speak to you? I think so.
That's very cool. Thank you so much.
So Dr. Venter is going to actually be in the lobby, and most of you have a program. If you did not get a program, please grab one on your way out. And he will be there to sign and autograph your program and to be out there for you. Also, please look at the Daily Journal today. If you want to pick up the Daily Journal on page 4, there's a story about Dr. Venter visiting you here at Mills. And students, I want to thank you for being part of history here at Mills and being here to listen to Dr. Venter and to see him receive this plaque. And hopefully it's a story that someday you'll be able to tell your student, your child, your family as they join us here at Mills. So thank you, students, for your participation, and teachers.